Today we have the topic nervous system, and it is very complicated on the one hand, but on the other hand I guess that anatomy and physiology of this system are much more complicated than the histology. Or it is better to say they have more practical application for future doctors. Why? Because first of all, neurons with the same structure but located in different regions of our brain, they might be responsible for quite different functions. That's why sometimes it is better to understand in which region this neuron is located than what structure it has. Okay, so location is more important than the structure. Then the second reason why I consider that histology knowledge has the secondary role in this system, that's because biopsy is very rarely obtained. Of course, because nervous system doesn't regenerate well, and because it is responsible for our memories, so only rarely we obtain a part of brain, only in case of tumors. That's why a histological structure of this system is important to understand how it works, but has less practical application than physiology and anatomy. But in any way, we concentrate today our attention on the most complicated structures where different cellular subtypes are located, and it is very important to understand the basics of their physiology. Okay, let's start from the most complicated structure, cerebral cortex. It has 6 layers, and there are no exact borders between them, so they are more or less conditional, and each layer differs by the cellular composition. So cells differ by their shape, but they differ more significantly by their function. So which layers do we consider? First of all, the most superficial is called molecular. Molecular because it is almost acellular. Only a few cells are located there, and they are called horizontal neurons. And in this diagram, we can see the distribution of cells in every layer. And we can see that layer 1 is almost devoid of cells. That's why it is called molecular. Easy to remember. The second layer is called outer granular. Granular because cells, they are very small, they have large nucleus, and those cells are resembling granules. That's why outer granular cells, and there we can find stellate pyramidal cells, and they are highly concentrated like granules. Then outer pyramidal layer containing pyramidal cells respectively. And this is the third layer. Here we can see, but they are small, and on this magnification we can't see exactly that their bodies are represented by triangular structures. Then inner granular layer, easy to remember because as we have outer granular, so respectively we have inner granular. Yeah, inner granular layer also concentrates stellate cells inside, but as I have already said, the shape is the secondary comparing with the function. Then ganglion cell layer, that's exactly what you have to remember, because this is the most functionally important layer with giant pyramidal cells. They are also called bed cells. Besides beds, he worked in Ukraine, in our capital, in the Kiev University, and he discovered these cells. As you can see, they are the largest, and they are 3 times larger than cells of the outer pyramidal layer. They are up to 120 micrometers, and they send very large dendrites which span all the above layers of our cortex. They reach the molecular layer even, so they collect the information from all those layers with the help of their dendrites. And they have a very long axon, which is not shown in the diagram, and this axon it is directed to the white matter, to the subcortical structures. So those neurons are motor, and they make the decisions, analyzing the information in our cortex. Then multiform cell layer is called so because it contains cells of different shape, multiform, multiple forms, multiple shapes. So multiform cell layer is the deepest one. So taken together, we have to say that what is especially important, that granular layers are responsible for sensory functions. So they get the information, they analyze the information, while pyramidal layers are responsible for motor functions. The difference is that, as you already know from physiology and anatomy, that sensory layers, they accept the information, while motor function means that neurons send the information from the cortex to the subcortical structures. And respectively, if we take the brain cortex from different regions, in those regions responsible for motor functions, pyramidal layers would be more developed, more thick, more prominent than granular layers. And some regions of our cortex, they are responsible for sensory function, yeah, for example, for our vision or hearing. Those regions, they would contain more developed granular layers than motor neurons. So the interrelationship, the relative thickness would differ depending on the region. Okay, let's move on. Here the specimen is given. I want to say first of all that this is the rat cortex, not human, but it shows us that really those layers are not equally developed and there are no exact borders. But let's try to distinguish the basic structures. Here I want to remind besides that you have to analyze and recognize specimens during exam, during module. So be attentive. If you want to get a good mark, you need not only to recognize but also to distinguish structures inside the specimen. First of all, what can we see? These are the giant pyramidal cells. Notice here they are, and they have triangular shape on the cross section. These are the bat cells, and they are located in this fifth layer in the inner pyramidal layer. Then also pyramidal cells could be found here. They are much smaller, and they correspond to the outer pyramidal layer. If to be honest, I'm not sure that rats, they have so complicated cortex as humans, but something like that. So we can see here the outer pyramidal, inner pyramidal. Between them we suppose the inner granulum. Above the outer pyramidal we're supposed to have the outer granular layer. And after that, so distinguishing only those two layers, we can more or less exactly distinguish others. Multiform cell layer should be under the giant pyramidal cells, and the superficial layer is molecular, and cell are very rarely found there. Okay, let's move on. Then, except unequal distribution of cells in our cerebral cortex, fibers are also organized in different way, and we can see that in the molecular layer, so-called tangential fibers are located. They correspond to the processes of horizontal neurons. There are two also bacterial stripes. One bacterial stripe between the molecular and outer granular layer here, and two layers of stripes of Berger, which correspond to the inner granular layer and inner pyramidal layer. So here they are. They could be found only in case we stain specimens for the myelinated nerve fibers. So in a conventional histological specimen is here, we can see those fibers. If to summarize, our cerebral cortex, it has 6 layers according to the cellular structure, and also it has an equal organization of fibers. So fibers are oriented in a vertical direction, in the horizontal direction, and those with horizontal direction could be found on the level of respective layers, which are given in this slide. Then, as I've already said, in the brain, in the brain cortex, different regions, they are responsible for quite different functions, and some of them, they have motor functions, some of them sensory and motor, they are responsible for different organs, and that's the subject of physiology. On the histological level, there were many attempts to find a so-called morphofunctional unit of the cerebral cortex. The unit is in which the unit of information is analyzed. And nowadays, the most popular idea is the brain column, brain barrel, or brain module. And here this concept is, it is believed that in our cerebral cortex, bed cells are the most important, and each bed cell represents the core of the brain module. Here the confocal image is given, three-dimensional histological image with bed cell and its branched dendrites. As we have said, they span all the above layers of the cortex. So one bed cell together with assisting neurons and fibers, they represent the morphofunctional unit which is called the brain module or column. What it includes? This unit includes one thalamic fiber coming from the thalamus. And you know the thalamus first, it receives and collects the information from all our organism receptors and then sends this information to the cortex. So one thalamic fiber with the afferent information comes to the cortex and span its 6 layers. Then 2 cortical fibers accompany it. Cortical fibers, these are the axons coming from another pyramidal cells, from the same or another hemisphere. So in the center of this cylinder, there are 2 types of fibers, thalamic and 2 cortical. And they bring the information which is to be analyzed. After that, this information is affected by a group of inhibiting and excitatory neurons. Some neurons, they accelerate, they augment this impulse. Some other neurons try to inhibit it. And if the information worth to be analyzed, if the information is really important, then it would excite pyramidal cell, and eventually pyramidal cell would send the impulse with the help of its axon. If the information is weak, unimportant, and considered as the secondary, if, for example, right at this moment we have much more important information to analyze, then such impulse will be inhibited. So we can't excite all pyramidal cells simultaneously. The brain will be overloaded. And as you know from physiology department, we need some inhibitory impulses to distribute the activity of our brain. So each moment of our life, our brain analyzes only the most important information right now. So what do you have to know? That brain module includes excitatory and inhibitory neurons. Excitatory neurons, these are the focal and diffuse types of stellate cells. Spiny stellate cells, okay, at least that. Stellate means star-shaped. And inhibitory neurons, which try to inhibit, these are the cell with axonic tuft, basket cells, exaxonic cells. And there is a group of neurons inhibiting inhibitory neurons. So they uninhibit. As a result, they have uninhibitory effect because they inhibit inhibitory. Okay. So these are the cells with double bouquet of dendrites. They have this name. Okay, to summarize the information, our brain cortex could be divided into functional units like dots. Each unit is called brain module, brain barrel, or the brain column. And in the center of this column, there are 2 fibers, thalamic and cortical fibers. Actually 3, but of 2 types. So those fibers, they bring the information to the column. And on their way, they interact with dendrites of pyramidal cells and 3 groups of neurons, inhibitory, excitatory, and those which inhibit inhibitory. So if information which is brought with the help of those fibers is important, then eventually it excites the pyramidal cells and they consequently activate subcortical centers. If information is considered as unimportant, weak, and if there is another center of excitation in the brain with more important, strong impulses, then inhibitory neurons inhibit this impulse and eventually pyramidal cells that don't do anything. And in this way, our brain cortex works. Okay, this is in general. And as you can see here, this diagram is given. Many columns exist, and in the center of each column there are fibers and pyramidal cells. Pyramidal cells, they send axons to another column in this hemisphere or another hemisphere. So in this way, we make decisions based on analysis of many input signals. Then next, cerebellar cortex. So cortex found on the surface of cerebellum has quite another organization, and it has a lot of cells which are organized only in 3 layers. So those 3 layers are very well distinguishable, very easy to distinguish them. So let's look at the specimen first of all. The outer layer is molecular, and it contains only 2 types of neurons, basket cells and stellate cells. Here their nuclei are, and it is called again molecular because comparing with other 2 layers, it has only a few cells. Next, Purkinje cell layer, the middle one, it contains a single layer of giant piriform cells, which are called Purkinje cells. Those Purkinje cells, they send branched dendrites to the molecular layer and a long axon to the white matter, to the subcortical nuclei. Then granular layer, it is called so because there are many cells like granules here, and it contains Golgi cells and granular cells, which is very easy to remember. Okay, this is in general histological structure and cellular distribution. Let's investigate the interrelationships between those cells. And before doing that, we have to say that as in the brain cortex, impulses are brought with autonomic fibers or cortical fibers. To the cerebellar cortex, impulses are brought with the help of climbing fibers and mossy fibers, which arise from different centers like inferior olivary nuclei, pontine nuclei from the pons, and after that they analyze the information which is brought is analyzed by cells in the cerebellar cortex. Why they are called climbing and mossy fibers? Because mossy, like a moss, they are very short and they reach only the deepest granular layer. They can't reach Purkinje cells or moreover molecular layer. At the same time, climbing fibers, they climb up to the molecular layer, and that's why they are called so. Okay, let's analyze this diagram, and it is very helpful and very easy to understand, I think. So our cerebellar cortex, it has 3 layers: molecular, Purkinje, and granular. First of all, we look at the fibers bringing the information. There are 2 types of fibers, mossy and climbing. As I've already said, mossy fibers, they are short as moss is, and climbing fibers, they climb up to the molecular layer. Then, first of all, we have to say that almost all neurons shown here, they have inhibitory action on each other. So almost all are inhibitory, except 3 elements. Excitatory impulses are brought with the help of mossy fibers, climbing fibers, and granular cells. All others are inhibitory, including Purkinje besides. Then, let's analyze the impulse. Let's trace the impulse which is brought with the help of mossy fibers. So mossy and climbing fibers, they bring information to the cerebellar cortex from our joints, muscles, vestibular apparatus about the location of our body in a 3-dimensional space. And you know that cerebellum in general, it is responsible for our locomotion, for our balance, equilibrium, and acts. So mossy fibers, they bring the information, excitatory information, and then transmit this impulse to the granular cells. Here it is of green color. Then granular cells, they send their long axon to the molecular layer, which it is split into 2. It splits into 2 via the T-shape manner, and this axon excites the Purkinje cells. Okay, so mossy fibers, they excite granular cells, which in turn excite multiple Purkinje cells in a molecular layer. What is important to say is that one granular cell is in contact with many Purkinje cells. So the impulse which is brought by mossy fibers is it has weak effect but excitatory effect on many Purkinje cells. Another aspect we have to highlight is that the synapse between the mossy fiber and granular cell is called glomerulus. So if you are asked what do you know about glomeruli of the cerebellar cortex, you should say that this is the synapse between those two. Then, what about the climene fibers? Climene fibers, they are coming to the molecular layer and directly stimulate dendrites of Purkinje cells. So climene fibers, they have strong excitatory effect on a single Purkinje cell, comparing with mossy fibers, which have weak effect on multiple Purkinje cells. Okay, and they are called climene because they climb along the dendrites. You know, dendron means tree. That's why this comparison is very helpful, because really climene fibers, they climb along the branches of dendrites of Purkinje cells. Then, what another cells could be found here? First of all, Golgi cells located in the granular layer. Golgi cells, they are interneurons, and they have their axons and dendrites directed towards the synapses between the mossy fibers in granular cells and axons of granular cells. So they are put between those two, and they provide negative and positive feedback regulation on this level. Positive and negative feedback regulation. This is the most important function. They have inhibitory action, I remind. So they also could participate in the formation of those glomeruli. But glomerulus means the synapse between the mossy and granular cell. Okay, then what about the molecular layer? There are two types of cells, as you remember. These are the stellate cells. Stellate means star-shaped, really like stars in the sky. They are scattered in the molecular level, and they are interneurons, and they have inhibitory action on Purkinje cells. The last cell type here is the basket cell, which is of blue color, and as well this is the interneuron with its inhibitory action on Purkinje cells. And the question is why they are called basket. Because they are axons, they wrap around the body of Purkinje cells and form something like a basket in this way. So if we look at the specimen, we can see really that around the Purkinje cells, those black fibers are wrapped, and exactly these are the axons of basket cells. These are so-called baskets. So if to summarize the information about the cerebellar cortex, we have to say that information is brought via mossy or climene fibers. This information excites Purkinje cells. At the same time, Purkinje are negatively affected by stellate and basket cells, and eventually they make a decision whether to be excited or not. And in case they are excited, they send inhibitory impulse to the subcortical, deep subcortical nuclei. In case Purkinje cells are inhibited or inactive, they uninhibit deep cerebellar nuclei. And in this way, decision is made. So the cerebellar cortex is much better understood than the cerebral, of course. Okay, I think we have considered almost everything. Let's move on. And the next structure to be analyzed is the spinal cord. Okay, in the structure of the spinal cord, we can distinguish easily the gray matter and white matter. Gray matter is concentrated in the center in form of butterfly, with its posterior horns and anterior horns. Sometimes lateral horns are present in some segments of our spinal cord. So the white matter surrounding it contains fibers but not cells. I mean glial cells but not neurons. And the gray matter is represented by the concentration of neuronal bodies. So bodies, they form nuclei, and which nuclei could be found inside the spinal cord. In the dorsal horns, there are two pairs, proper and thoracic nuclei, nucleus proprius, nucleus thoracicus, and as well two substances, gelatinous substance and spongy zone. Those structures can be distinguished in the posterior horn. Then in the lateral horn, nucleus intermedio lateralis, intermedio medialis, lateral and medial, respectively, and in the ventral horn, very easy to distinguish 5 nuclei. They are called depending on their location. So dorsomedialis, ventromedialis, dorsolateralis, ventrolaterales, and the central. Those 5 present there. Of course, we can distinguish them on a specimen. So if we look at the specimen, what can we find here? Of course, we distinguish white matter from the gray matter, the central canal filled with the cerebrospinal fluid, and ventral horns. They always can be distinguished from posterior because they are much more broad. And there are multipolar neurons, also very well distinguishable. Of course, they are not grouped in nuclei, so we don't ask you to distinguish those nuclei because it depends strongly on the level on which the specimen was obtained. Then, talking about the spinal cord, we have to mention those structures accompanying it, which are spinal ganglia or sensory ganglia. They are located close to the dorsal side of the spinal cord, inside the dorsal roots. So now one more term should be mentioned. We have dorsal horns, and they continue into dorsal roots. And also we have ventral horns of the spinal cord, which continue into ventral roots. So please do not confuse the horn and the root, because horn is inside, this is the part of gray matter, root is the part of white matter, and it is outside. As dorsal horns and roots, they are sensory. Ventral horns and roots, they are motor. And as spinal ganglia are located inside the dorsal root, they are sensory, and they are used to deliver the information to the nervous system. So roots and horns, they differ by the direction of nerve impulses. Through dorsal, the information enters the nervous system. Through ventral, the information is sent from the nervous system to the peripheral organs. Notice that eventually those roots, they fuse with the formation of the spinal nerve. So inside the spinal nerve, we have both afferent and efferent neurons combined. But coming close to the spinal cord, they split into ventral and dorsal, incoming, input and output respectively. Let's investigate the structure of the spinal ganglion in details. First of all, it contains, of course, neurons, and please remember that there are pseudo-unipolar neurons. This is the typical structure and typical place of their location. Pseudo-unipolar means they have one process which further splits into 2. So like unipolar, but actually it is bipolar. Then each neuron is surrounded by mantle cells and satellite glial cells. Those satellite glial cells, they are the part of our glia cells. If you remember previous lecture about nerve tissue, glial cells in the central nervous system, they are divided into macro, micro, and in the peripheral nervous system, they include satellite cells, Schwann cells, and some others. Okay, so satellite cells, they are similar with astrocytes in the central nervous system. Okay, and neurons, they contain very well developed nissl substance, which is the evidence of their activity, which reflects the degree of their activity. So spinal ganglion is represented by those 2 types of cells, pseudo-unipolar neurons with large bodies surrounded by the satellite glial cells. Then notice what we can distinguish in the specimen: the dorsal root, which is here, and the ventral root. Together they fuse and form the mixed nerve or spinal nerve. So what we ask you to distinguish in the specimen, those 2 types of cells. Then you have to distinguish dorsal root from the ventral, because spinal ganglion is inside the dorsal sensory with afferent impulses, afferent nerves. And another root is respectively ventral or motor. Together they form the spinal nerve. Okay, let's move on. This is the specimen, and we have discussed it just. So next, this diagram showing us the chain of neurons. So the information which is brought to the spinal ganglion is transmitted from one neuron to another, and respectively these pseudo-unipolar neurons, they send the information to the dorsal horn. So this is the direction of the impulses. And spinal ganglia, they derive from the neural crest. Please remember that. So they derive not from the neural tube, but from neural crest. Then, as for the autonomic ganglia, sympathetic and parasympathetic, they belong to the autonomic nervous system, and they have almost the same structure, except neurons are multipolar in this case. And look at the specimen here, multipolar neurons and satellite cells around them. The difference between the sympathetic and parasympathetic is that sympathetic are more close to the central nervous system, while parasympathetic close to the peripheral organs. Parasympathetic ganglia could be found inside our intestines, respiratory system, and many peripheral organs. Okay, besides, notice that inside lipofuscin granules could be found, and this is the sign of aging. They are deposited with aging inside. Okay, then sensory receptors. Besides, this is the part of your lecture about the skin, and I want to remind one more time about those peripheral receptors, predominantly in the skin, but also found in other organs. Why? Because the Nobel Prize in this year was awarded to two scientists investigating exactly mechanoreceptors and thermoreceptors found in our skin particularly. So they are considered as very important because they are responsible also for pain, and they are very intensely now investigated to help patients to relieve the pain as well. Okay. Then embryonic development of the nervous system, and now I want to return to the embryology lecture. And you remember that early in embryonic development, our ectoderm forms the neural tube, and the closure of the neural tube is called neurulation, when plate forms the tube, and folic acid is especially important in this process, right? Then, after its closure, in the anterior part of the tube, 3 vesicles appear. And this diagram is very helpful. I think I don't need to explain, just remember what is written here very exactly. So 3 vesicles are formed at the beginning. The middle one, or mesencephalon, midbrain, gives rise to the mesencephalon, respectively, while others, 2, they have more complicated development. So anterior vesicle, or the forebrain, gives rise to 2. The telencephalon and diencephalon. Besides diencephalon, this is the place where eye cups are formed. This is the topic of the next lecture about the eye. And the rhombencephalon, or the hindbrain, the posterior brain, divides into metencephalon and myelencephalon. Myelencephalon gives rise to medulla oblongata, while metencephalon further splits into pons and cerebellum. Okay. So that's what you have to remember about the brain. Then, if to investigate its histological structure, we have to highlight that in the dorsal region of the nerve tube, predominantly sensory neurons are found, while in the ventral regions, motor neurons are formed, like in the spinal cord, yeah. And so-called alar plates are formed in the dorsal regions and basal plates in the ventral regions. So these are the concentration of sensory and motor neurons, respectively. And then, except the specification, dorsiventral specification, we have to say that there are 3 layers in our nerve system, nerve tube. The innermost line in the cavity, this is the neuroepithelial layer, which eventually gives rise to ependymal cells. Then the middle layer is mantle, and it concentrates neurons, and it is responsible for the gray matter formation, while the outer layer is called marginal, and it is responsible for the white matter formation as it contains predominantly cell processes. Okay, so please remember 2 plates and 3 layers. After that, in every compartment of our brain, alar and basal plates are not equally developed, and in more modern, in evolutionary more developed regions, the alar plates or sensory neurons, they are more developed. They develop more intensely. So in the higher centers, alar plates are more developed than basal plates. Sensory neurons prevail over the motor. What is also important is to say that along the longitudinal axis, those brain vesicles are specified due to the consequent expression of homeobox genes. We have already mentioned those genes in the lecture about embryonic development, digestive system, because along the segments, along the longitudinal axis, different segments, they have different specification depending on the type of homeobox gene which is expressed there. So this is a very ancient family of genes, and they perform very important function in the development of different organs, of course, in the specification of brain. So along the longitudinal axis, specification of each brain vesicle depends on homeobox genes. After that, sensory and motor neurons are formed in the neural tube under the influence of the ectoderm and cord respectively, notochord respectively. So epidermis, or the ectoderm above the neural tube, expresses a group of factors helping to specify the dorsal neurons into sensory one, while notochord expresses another group of factors helping to specify ventral region of the neural tube into motor neurons. And those factors, they are very important, of course, in the development. Then the last aspect of the embryonic development we have to mention is that at the beginning of the development, neural tube is represented by only a single layer of cells. After that, it splits into several layers, and the innermost transforms into ependymal cells. And those ependymal cells, they have very crucial role in the embryonic development of our central nervous system, because in embryonic development, at their base, they have a very long process which extends to the outermost layers, and along those processes, other cells migrate and form layers of our brain cortex, for example. So if those processes are not properly formed, then the cytoarchitectonics of our brain cortex will be affected. So ependymal cells, they send this long process, and first wave of migration results in the formation of the most deep 6th layer of the brain cortex. After 6th layer is formed, then cells migrate through them to form the 5th layer. After that, 4th layer is formed, and so on up to the molecular layer. So the deepest layers are formed at the beginning, while the superficial layer at the end. And in the process of their migration, ependymal cells, they play the pivotal role, because in the embryonic development they form those routes for migration, those axial processes which are used at the scaffold for migration. And you know that in the region of the third ventricle, ependymal cells, they still preserve those processes, and they communicate the cerebrospinal fluid with thalamic nuclei, helping thalamus to get the information from the cerebrospinal fluid about the concentration of some nutrients. That's the subject of the previous lecture. Okay, this is the very complicated aspect of the central nervous system development, and this is the last slide. So thank you for the attention, and have a good one.